All right, welcome back. Today we're going to be talking about macromolecules, which are going to be the building blocks for all of life. So, before we can start talking about this, we have to be talking about the chemistry of life because we're in biology, we're learning about life. We need to know what are the foundations of life. So, organic chemistry itself is the study of carbon-based compounds. So organic chemistry is the study of carbon-based compounds. And organic means living. All living molecules are based in carbon. Everything living contains carbon. Therefore, carbon is going to be essential for life because all living things contain the element carbon. Now, this is going to be very, very important for the different macromolecules that make up living things because carbon can form long, complex chains and rings. These complex chains and rings are going to form the macromolecules that are found and essential for life. Therefore, we are talking about the macromolecules, which are essential for life. So, small organic molecules join together to make large organic molecules called macromolecules. So, small organic molecules, very small ones, are going to join together to make larger, more complex molecules called macromolecules. Now, there are 4 macromolecules that we are going to be talking about. The first one is carbohydrates, followed by lipids, and then proteins, and nucleic acids. Again, all found in living things. So earlier we said small organic molecules join together to form large molecules. These small organic molecules are building blocks called monomers. Prefixed mono means one. So one building block, one subunit, is going to form with other monomers. So many monomers come together to create polymers. And the prefix poly means many. So many monomers form a polymer, which are going to be known as the large macromolecules. So carbohydrates, lipids, proteins, and nucleic acids are polymers because they are made up of many monomers joined together. The way that we build polymers is by a chemical reaction known as dehydration synthesis. Dehydration means you take away water, and synthesis means to make. So essentially, you are taking away a water molecule to form a bond. So dehydration synthesis takes away a water molecule to form a bond. This process is going to require the input of energy and the assistance of enzymes. So again, you take away a water molecule to make a bond, dehydration synthesis. Take away water to make a bond. Now, sometimes the opposite has to be done. Say we eat food, when we need to break down our food. We have to break down the polymers that we take in, and this is going to involve a reaction called hydrolysis. Hydrolysis is the opposite of dehydration synthesis. In this one, you add water to break a bond. So take to make. Let's write that down. Take to make. Add to break. So you add water, break a bond. So, what is the difference between dehydration synthesis and hydrolysis? Dehydration synthesis, you take away water to make a bond. Hydrolysis, add water to break a bond. I had to pause for a moment there because my battery was dying. Okay, so moving on. We just answered that question. Now we're going to start looking at each of the macromolecules individually, and we're going to start with carbohydrates. So here's an image with some of the lovely carbohydrates in the world around us. Essentially, carbohydrates are anything and everything made of sugar. So carbohydrates contain 3 elements: carbon, hydrogen, and oxygen. So C for carbon, H for hydrogen, and O for oxygen. Now the monomer, or the building block of carbohydrates, are called monosaccharides. Mono means one or single. Saccharide means sugar. So a monosaccharide is a single sugar. And the cool thing about monosaccharides is the breakdown of these elements found within the carbohydrate. They're always found in a 1 to 2 to 1 ratio. Meaning, here's an example here. This is a sugar known as glucose. Glucose has 6 carbons and 6 oxygens. Those are our 1. The 2 in the middle is going to have twice as many carbons, 12 hydrogens. So if we did this on the side and I wanted to, let's just make up our own carbohydrate. We're going to have carbon, hydrogen, and oxygen. Carbon and oxygen are always going to be equal. So if we had 7 and 7, carbon and hydrogen are always equal. Hydrogen is going to be double the amount of carbon. Another thing that you could also think about, carbon and oxygen are equal. You add them up, you get hydrogen. So it's just an easy way to remember it. Carbon and oxygen are equal. You add them up and you get your number of hydrogen. It's a 1 to 2 to 1 ratio. Now, the function of carbohydrates in our bodies, or in living things in general, are to provide quick energy, so again, I said that carbohydrates are sugars. Little kids eat lots of sugar. They get quick energy. It stores energy short term. So short term energy storage. And it also provides structure in cells. The cell wall in plants is made up of cellulose, and the cell wall is for structure in plants. Any starch is found in plants. And in humans, the carbohydrate that we have is called glycogen. Now, typically, when we're looking at sugars, the more common sugars are going to end in the suffix "ose." So anything that ends in OSE is a type of sugar. So glucose, fructose, sucrose, maltose, all of those end in OSE and therefore are sugars. Sugars can also be simple or complex. So a simple sugar would be a monosaccharide. Again, a monosaccharide is a single sugar. An example would be glucose. It looks like this. It just has one ring, single sugar. In the middle, we would have a disaccharide. The prefix di means two. So a disaccharide is two simple sugars linked together, and the example here is sucrose. And then a complex sugar would be called a polysaccharide. Again, poly means many. So when you link together many sugars, you get a polysaccharide, and an example of that would be starch. And again, the types of reactions that we talked about are used to build or break any of these macromolecules. So dehydration synthesis is used to build a polysaccharide, and hydrolysis is used in our bodies to break them down. So again, those reactions apply to all of the macromolecules we're going to talk about. Just a bit more about polysaccharides specifically. They are typically used for energy storage and structure in living things. So starch is used to store energy in plants. One way I remember this is because when I think of starches, I think of like potatoes. Potatoes are really starchy. And potatoes are found underground. They're actually tubers. And it's the storage form for the plant. So that's how I remember starch. Potatoes are starchy. And it's found underground for storage. Energy storage in animals happens through a polysaccharide called glycogen. So in our bodies, we store sugar as glycogen. Another common sugar is called cellulose, and it makes up the cell wall in plants. And again, the cell wall is used for structure and support. And the last one that you probably haven't heard of, but you're familiar with, is a polysaccharide called chitin. Chitin is found in any type of organism that contains an exoskeleton. So a lobster, a crab, crayfish, the ones that have that hard outer shell, that shell is made up of chitin. And it's also found in organisms like fungi. So it's another example of a polysaccharide. So our next category of macromolecules are lipids. And you're familiar with lipids because lipids are essentially fats. Lipids are composed of the elements carbon, hydrogen, and oxygen. And these are going to be long hydrocarbon chains. Hydrocarbon means carbon and hydrogen. So if you were to look here, this is a bigger image here. Long chains, so these whole end, I keep forgetting I can't write on the PowerPoint. The part that's highlighted in yellow is primarily carbon and hydrogen. There's only a few oxygens on here in a few places. So lipids are mainly carbon and hydrogen with a little bit of oxygen. Now, the monomer or the building block of a lipid is called a triglyceride. A triglyceride is a fatty acid. One way that you can remember this, we have a little memory trick over here for you. The memory trick for fatty acids and lipids are keep your lipids off my fatty acids. Keep your lipids off my fatty acids. And the function of lipids in living organisms is to store energy. So energy storage oftentimes happens in the fat. Now, lipids store much more energy than carbs because this is long-term energy storage, not short-term, but long-term. Lipids also used to cushion organs, so protection of organs, and it insulates the body. That means keeps your body warm, like whale blubber would keep whales warm. Some properties of lipids. They are nonpolar and hydrophobic, meaning they don't like water. So we talked about in our last video about the properties of water, and we said that anything that is not polar is hydrophobic, therefore does not mix with water. So anything that does not mix with water is going to fall in the category of hydrophobic and is more likely than not going to be a lipid. Examples of these are fats, oils, waxes, steroids, and in our cell membranes, phospholipids. And the structure of a lipid monomer is a glycerol molecule linked to a fatty acid. And lipids do come in two categories. Lipids can be either saturated or unsaturated, and I'm sure that you guys have heard of these as far as relating to health. So saturated fatty acids or saturated fats contain no carbon-to-carbon double bonds. And what this is going to do, if there's no double bonds, a double bond forms a kink. So over here you see a double bond. It forms a kink or a bend in the fatty acid. Therefore, saturated fatty acids, no double bonds, it's going to be perfectly straight. And if they're perfectly straight, they can pack together, and therefore they are going to be solid at room temperature. Anything that's going to be solid at room temperature and is perfectly straight and can pack together, These are the ones that can contribute to cardiovascular disease because these fats can clog arteries. Therefore, they are less healthy for you. Examples of these are animal fat, so fat in animals' bodies, but also fat found on like steaks and meats, as well as things like coconut oil. So for cooking purposes, anything you're going to put inside of your body, coconut oil is less healthy. And you know that coconut oil is a saturated fat because when you buy it, it's not a liquid. It's a nasty, white, chunky type of solid. And that's how you know that it is saturated. An unsaturated fatty acid has carbon-carbon double bonds, at least one. And that means it is going to be a liquid at room temperature. The fatty acid itself is bent. It has a bent shape. Examples are going to be plant fats and fish fats, as well as things like olive oil, because they are liquids at room temperature. And these are going to be the healthier types of fats. And one last thing about fatty acids and lipids before we go. We had mentioned phospholipids being found in the cell membrane. So a phospholipid is composed of a phosphate head, I put one extra T in there, a phosphate head with two fatty acids at the bottom. And these make up cell membranes in living things. Right, moving on to category 3, proteins. Proteins are, we have images over here, all of our meats, dairy, eggs, all contain protein, as well as beans as well. That one is not on there. Proteins not only are found in food, but it can also be found within our bodies to help with reactions. So proteins are going to be very important. So you guys know fireflies glow. A protein is actually what makes them glow. So it does many different things in all types of organisms. In poisonous organisms like the pufferfish, scorpion, and king cobra, a protein is the neurotoxin that makes organisms sick if they get bit or stung. So proteins do more than just provide us food, including spider webs has a protein, our skin, our nails, bird beaks, they're all made of different types of proteins. So proteins themselves are made up of carbon, hydrogen, oxygen, and also nitrogen. And proteins are made up of the monomer or the subunit amino acids. And we do have a memory strategy to remember this one. Pro-amino. It rhymes. Pro-amino. You can remember protein amino acids. In all living things, there are 20 different types of amino acids. Proteins are going to be long chains of amino acids, and each protein has a different number of amino acids and all put together in different orders. So therefore, proteins can do many different things and have many different structures. Some functions and examples of proteins. Proteins can be used for structure. For instance, we talked about back a couple slides, we talked about hair, skin, and nails scales, as well as tusks and beaks. Um, Hormones like insulin are also proteins. In our muscles, proteins are used for movement. They have a big role in our immune system to help protect us against germs. The very biggest one that we're going to talk about for proteins are enzymes. We're actually going to have a whole nother presentation on enzymes themselves. And enzymes speed up chemical reactions. Therefore, they are very, very important in living things. And proteins also have a function in transporting nutrients throughout our bodies, like the hemoglobin protein that carries oxygen to the rest of our bodies. So proteins are essential in living things. So looking a little bit at the structure of proteins. So again, proteins are made up of amino acids, pro-amino, and long chains of amino acids make a polymer. Each amino acid is linked to the next by a peptide bond. So peptide bonds are only found in proteins. Another thing that you'll notice about amino acids is all amino acids have the same basic structure. So meaning we have 1, 2, 3, 4, 5 different amino acids. And above the letter R, they are identical. The thing that makes each amino acid different is this R group, which is the variable group, the part that differs between one amino acid and the next. And that's what's going to give each amino acid its function. The structure of the entire protein itself is dependent on the amino acids inside of the protein. Depending on the amino acids, where they are in the protein, it's going to make the protein fold and twist a specific way, giving a protein its 3D shape. So at the very surface, a protein is made up of a chain of amino acids found here like beads on a bracelet. Then these amino acids are going to interact with each other, and it could give the protein either a helical shape or a flat shape. Sometimes different protein chains will interact with one another. So here we have a blue protein and a red. Then they will fold upon themselves again to create the protein itself that is going to do a job. So there are many different levels to protein structure. And in proteins, the structure of the protein is going to determine its function. Proteins are very, very specific. So proteins are specific right here, meaning one protein only do a certain job. They have a specific shape to do their specific job. Anytime a protein is unfolded or destroyed, it is called denaturing. The denaturing of a protein means it can no longer do its job. So if a protein changes its shape at all, it no longer works because proteins are highly specific. A few things that can change proteins are temperature and pH. So one example of a protein is an egg. When you crack an egg, I mean, everybody knows what an egg looks like. It has the yellow yolk in the middle that is liquidy, surrounded by a clear-ish, kind of like jelly-like fluid. However, when you put that egg in a pan and you heat it up, the egg completely changes. That is an example of denaturing. When you see that change in the egg, you have denatured the proteins in the egg and it can no longer do whatever it was supposed to do. These changes are irreversible, so you can't go back. You can't uncook an egg. Similar thing happens with pH. Something very, very acidic is essentially going to burn whatever it comes into contact with. If you were to put acid on an egg, it would look very similar to if you heated an egg, because it denatures the proteins inside of the egg. Okay, in our to talk about our nucleic acids. Nucleic acids are made up of carbon, hydrogen, nitrogen, oxygen, and phosphorus. And the building block of a nucleic acid is a nucleotide. The way that you can remember that nucleic acid goes with nucleotide is that they both start with the same letter. So nucleic acid and nucleotide both start with the letter N, therefore they go together. The function of a nucleotide is to store and transmit hereditary information. So the passing on of traits from one generation to the next. And this happens through two molecules, RNA and DNA. And with RNA and DNA, the N and the A stand for nucleic acid. So ribonucleic acid, RNA, when you can transmit hereditary information. DNA is deoxyribonucleic acid. Again, NA stands for nucleic acid. That is going to be a double helix. And it contains all of an organism's genetic information, and then that information is passed down to the offspring of that individual. So we set up here, nucleic acids are made up of nucleotides. Nucleotides have three parts. The first part is a base. It is a nitrogen base, meaning the base contains the element nitrogen. It has a pentose or a 5-carbon sugar. The sugar depends on the type of nucleic acid. DNA and RNA have different sugars. And it also includes a phosphate group. And we draw a nucleotide. I'm not sure if in this box. You guys will draw yours here, but just because I'm going to draw a little larger, I'm going to draw on the other side here. So a nucleotide has a pentose or a 5-carbon sugar, a nitrogen base, and a phosphate group. Your phosphate group is always a circle and it has a P. Sugar is pentose, meaning it has 5 points. And then the nitrogen base. The function of nucleic acids, again, is to pass on information. So it stores lots of hereditary information. And the code is found in the series of bases in the DNA or the RNA molecule. This is one building block. DNA and RNA is made up of many of these. So if we scroll up a little bit and look at this picture, there are many nucleotides found throughout a DNA and an RNA molecule. The sequence of bases is what changes. Therefore, it contains the information. It encodes information that is going to be passed from the parent to the offspring. And each unit of information is called a gene. And altogether, your genes make you who you are. And that is the function of the DNA and the RNA. So the last thing we have at the bottom are review questions. Review question number 1. What is an organic molecule? An organic molecule is a molecule that contains the element carbon. If it contains the element carbon, it is an organic molecule. Number 2, explain dehydration synthesis and hydrolysis. Dehydration synthesis is a chemical reaction that takes away a water molecule to make a bond. So we take away water to make a bond. Hydrolysis is adding water to break a bond. So hydrolysis, adding water to break a bond. Number 3, what are the 4 macromolecules of life? There are carbohydrates, lipids, proteins, and nucleic acids. So one more time, carbohydrates, lipids, proteins, and nucleic acids. And the last, what are the functions of each macromolecule? Carbohydrates, fast energy, structure. Lipids, long-term energy, insulation, protection of organs. Proteins speed up chemical reactions. They help protect you from bacteria and viruses. They transport nutrients throughout your body. That's just a few of those for proteins. And then nucleic acids, they're used for heredity. Okay, so there's going to be lots of practice on this on Canvas for you guys to go through to review the various macromolecules.